Hey everyone, welcome to our third video about enzymes. This time we're going to talk about active sites and how they get together with substrates, temperatures, pHs, like those two things can really have a big effect on enzymes, and just some examples of enzymes. First, let's talk about active sites. So, the active site, which we've talked about in our previous videos, is the part of the enzyme that is kind of like an empty space that pulls the substrate in and helps the reaction take place. It's actually the site of where the reaction takes place when an enzyme is involved in speeding up a reaction. But how do substrates and active sites actually get together? Well, there are two theories. The first is called the lock and key model. And in that one, the enzyme and the substrate already have the perfect correct shape to go together with each other. The second theory, which is a little more modern and kind of builds on some of the increased technology and knowledge that we have, is called the Induced Fit Model. And that is that when substrates approach the enzyme, it changes the shape of the molecules slightly to make them fit together. Let's look at some pictures because that's a lot of words that kind of describe what's going on, but a lot of people are visual. I know I am myself. So, this is the lock and key model. This is the idea that the enzyme's active site, this kind of empty space where the substrate will come attached to the enzyme to do the reaction. The lock and key model is the idea that this active site is already perfectly shaped to fit together with the substrate. So, in this like imaginary molecule shape, can you see where this active site is exactly the same shape as this substrate already, and it would already fit together really perfectly, and the reaction could take place, and then the product be released and the enzyme be reused? Well, that's the lock and key model, that it's already the perfect shape. But modern technology and advancements kind of have indicated to us that not all enzymes already have the perfect shape active site for their substrates. So we've come up with this induced fit model. And what you can see here is here's the enzyme and there's the active site, but it's not quite a perfect shape for the substrate yet. So what's going to happen is that when the substrate gets close to the active site, the enzyme will change shape very slightly so that the substrate can bind into the enzyme. Then the reaction takes place, the products are released, and the enzyme goes back to its original shape so that it can be reused. And it'll change shape again when a new substrate comes near. It'll change shape to grab onto that substrate, to bind to it, and then allow the reaction to take place. The products are produced, and then the enzyme reused. Here's a side-by-side look at that lock and key model and induced fit model. So like I said, remember lock and key model, the enzyme is already the perfect shape for the substrates. They attach or bind and they do their reaction, and then the enzyme is released to be reused and the product is made. In the induced fit model, which you can see down here, the enzyme is not the perfect shape for these two substrates, but when they get nearby, the enzyme changes shape slightly so that the substrates can fit there. Then, after the reaction takes place, the enzyme is released to be reused over and over, and the product is made. Either way, with either model, you get a similar product. It's just that these are two different theories about how enzymes are actually shaped. So, lock and key, it's already perfectly shaped. Induced fit, it's got to change a little bit when the substrates come near. All right, we've talked about the shape of the enzyme, how the active site works, how it speeds up reactions, but what effects do temperature and pH have on how enzymes work? Well, remember that enzymes are a special type of protein, and proteins can be affected by temperature and by pH. So, we've actually found out in scientific research that certain enzymes work better at certain temperatures, but there are some general trends. In general, when things get warm, they increase speed. When things get cold, they decrease speed. And when things are very, 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 very hot, that can inactivate or denature an enzyme. So you're going to hear me use this word denature throughout this module. And denature means inactivate or make it broken and make it not work. So if you have extremely high temperatures, like boiling hot, is going to cause the enzymes, which are proteins, to kind of come unfolded and not work anymore. And they won't be able to speed up reactions anymore if it's very, very, very hot. So here's an idea of looking at the effects of temperature on enzyme activity. You can see the ideal temperature for this enzyme is going to be about 40 degrees Celsius. And when it gets colder, the enzyme activity gets a little lower. When it gets warmer, the activity might get a little higher for a while, but when it gets too hot, that enzyme is going to be denatured and it's not going to actually work at all. Its activity will go all the way to zero when it gets too hot. Next, let's talk about how pH affects enzymes. We talked during the properties of water module about pH, and we said that living things need water to be the proper pH. And why did we say that pH matters to living things? Well, it's specifically because enzymes are super sensitive to changes in pH. If you have pH that is too acidic, it can denature or inactivate or break enzymes. If you have pH that is too basic, it can slow down or denature or inactivate or break the enzymes too. Let's look at some examples. So here are a couple of different types of proteins that are enzymes. There's pepsin, amylase, and trypsin. And what you can see here is that pepsin actually has the highest amount of activity going on at a pH that's about 2 or 4. Low pH, remember, is acidic. So pepsin, that might be in your stomach where there's a lot of stomach acid, and that pepsin is actually going to be able to work at those low pHs, and it actually wouldn't work at a neutral or higher pH. See, pepsin's activity is only at low pHs. Amylase is pretty different. Amylase works really well at a neutral 7 pH, and it does not work at a low acidic pH. And finally, trypsin. Trypsin's kind of interesting. It works at a slightly basic pH, a pH of 8, and it does not really work very well at acidic pHs. So all different enzymes have different pHs that they like or prefer. And if they're at a pH that is too far away from the pH that they prefer, they're going to be denatured or broken and not be able to make reactions happen faster. We're going to see this effects of temperature and pH when we do our lab this week. So look forward to that. You're going to actually see Larson in the lab video, so I think you'll like that one. All right, here are just some examples of enzymes, and these are just a lot of enzymes that are actually found in human bodies. There's lipase, which breaks down your lipids, like your oils and your fats, remember, from macromolecules. There's amylase, which is in your saliva and breaks down your starches into the smaller sugar monomers or monosaccharides. There's maltase, which is also found in your saliva or spit, and it breaks down sugars into glucose. So it takes your polysaccharides and makes them into monosaccharides. There's catalase, which is an enzyme that breaks down hydrogen peroxide into its components of water and oxygen. There's lactase, which helps break down milk's lactose. And there's protease, which helps actually break down old proteins during your digestion in your stomach. So think about this. What do all of those words end in? They end in ase. Most proteins, and enzymes are an example of proteins, will end in ase or in, like you saw, trypsin, pepsin, but a lot of them end in ase. So when in doubt, if you ever see like on a test or a quiz or even like the SOL test, if you see something that ends in ase, you can bet that it's probably an enzyme and that its job is to speed up reactions by lowering the activation energy. If this is a page that has a bunch of review, I'm going to post this in the supplemental and optional learning opportunities in Canvas. So if you need to see a summary of all the information from the past 3 videos, it's going to be there and you'll be able to look at it and even click the practice link to see a Quizlet that you can use to practice, because there were a good amount of vocabulary words in this module. Thanks so much for listening. I hope you've enjoyed learning about enzymes, and please let me know if you have any questions or need any help.